All right, hello everybody. Today I'm going to be going over our introduction to genetics. So the next unit, Unit 6, is on um, human health and the environment, and we're going to start off with heredity and genetics. So how genes are passed on from parents to young and how genetics work. Basically, you'll be getting into a lot more details of this in biology, but we do want to introduce you to it now so it's not all new to you when you get into that class. So I'm basically going to go through the PowerPoint that I've uploaded with you and explain it for those of you who need to hear it from another person. And if you have extra questions when you're working on your worksheets, please view all the videos I've uploaded into the playlist for this class and contact me via email if you have any questions. So starting off with genetics, the study of genetics began with observations from the scientist Gregor Mendel in the 1800s, and he noticed that his plants, his snapdragon flowers and his pea plants, although they were coming from the same parents, they had different traits. Some of them were purple, some of them were white, some of them were yellow. So he began to study how those traits got segregated or split up from the parents to form completely new traits sometimes. Now, he worked almost 150 years ago, so he was the first person to really start studying genetics, and his studies are what we base most of our knowledge on genetics today on. So moving into that, Mendel observed several traits in his plants, and he developed all these studies where he would isolate genes to make purebred plants and then cross those purebred plants to make hybrids. So some of the traits he noticed were seed shapes. Some of them were round, some of them were wrinkled, the color of the seeds, some of them were yellow, some of them were green, the flower colors that they produced, some were violet, some were yellow, some were white, and he would make purebred plants, cross them, and isolate these traits and see how they were expressed in the different plants. So let's talk about genes for a minute. What is a gene? A gene carries information that determines your traits. So genes are made up in your DNA of codes from nucleotide sequences. Again, you'll learn more details on that stuff in biology. And then traits are those characteristics you inherit from your parents, like your eye color, your hair color, whether you're tall or short. So your genes, which you get from your mom and your dad, combine together to determine what type of traits you have. And your genes are kept in your DNA, and your DNA is coiled up into these chromosome pairs that are in every single one of your cells. So genes are located on chromosomes, and chromosomes come in pairs that can then be split in half whenever your parents' bodies make new sex cells, sperm or egg. So chromosomes come in pairs, and there are thousands of genes on each chromosome. And the human karyotype, which is all the chromosomes that make up our DNA, we actually have 23 sets of chromosomes, so a total of 46 individual chromosomes. And half of the chromosomes come from one of your parents, and the other half of the chromosome comes from your other parents. So one from mom and one from dad. And they come together when the sperm and the egg cell fuse together in fertilization. And again, your parents got their chromosomes from their parents. So they got one from mom and one from dad, and that formed those chromosomes. And then those genes got split apart when they went to make you. Alleles. So for every trait, like eye color, hair color, or trait, you can have a variation, which is called an allele. So blue eyes, brown eyes, green eyes, these are all different alleles for the same trait. And you get one allele from mom and one allele from dad, and they can come together sometimes to give you the same phenotype. Sometimes you get a different phenotype depending on what the genotype is. So what is a phenotype and what is a genotype? Well, The genotype is the genes of an organism. So if you've got a dominant allele from mom and a recessive allele from dad, then you have a heterozygous genotype, meaning that your alleles from each parent are different from each other. If you have a dominant allele, say the blue-eyed allele from mom, the brown-eyed allele from mom and the brown-eyed allele from dad, if you got two dominant alleles from both of your parents, then you have a homozygous genotype, meaning that your alleles are the same from mom and dad. You can also have a homozygous recessive genotype if you got two recessive genes that are the same from mom and dad. The phenotype is the physical appearance given off by a trait. So your genotype and your phenotype can be different. You can have a homozygous dominant genotype or a heterozygous genotype and wind up with the same phenotype. But if you have a homozygous recessive genotype, then your phenotype will be different from that of the dominant genotypes. So, for example, if you look at slide number 15 on the PowerPoint, there are three different plants shown there. We have a homozygous dominant purple flower, a heterozygous purple flower, and a homozygous recessive white flower. So the phenotypes for those three flowers are purple, purple, or white. That's their physical appearance. The genotypes are homozygous dominant, heterozygous, or homozygous recessive. That is what their DNA code is that they got from the alleles from their parents. Dominant refers to a genetic feature that hides or dominates the recessive trait in a phenotype of an individual. And we always show dominant alleles with a capital letter. Recessive alleles are hidden underneath dominant alleles. So the only time we're ever going to see a recessive allele shown in the physical phenotype of an individual is if they have two recessive alleles from both parents. So for example, blue eyes is a recessive trait. So if you get one dominant brown eye trait from dad and one recessive blue eye trait from mom, then your phenotype is going to show brown eyes. But if you get two recessive alleles, you get a blue-eyed allele from mom and a blue-eyed allele from dad, then your genotype will show blue eyes because you are homozygous recessive. And we'll get some practice with this in with your worksheets and your Punnett squares too to make sure you really understand the difference between dominant and recessive and homozygous versus heterozygous. So segregation of your alleles happens when your parents are forming their sex cells during the process of meiosis, which forms gametes, that is sperm and egg. So the chromosomes from your parents will split in half, and one of them will wind up in one cell and one of them will wind up in another cell. This is why children from the same parents can have different genes, why siblings don't always look exactly the same, because those alleles get segregated and split apart when the sperm and egg are being created. So not all sperm cells coming from the same father are the same, and not all egg cells coming from the same mother are the same, because those genes from their mother and father got split apart during meiosis. Homozygous is a type of genotype where the two alleles are the same for a trait. So big R would be homozygous dominant, little r would be homozygous recessive. And heterozygous refers to when two different alleles for a trait come from the parents. So if you were to get one dominant from mom and one recessive from dad, say your genotype was big R, then that would mean you are heterozygous for that trait. And you are going to express the dominant allele in your phenotype. So there's some practice on your PowerPoint showing that we use dominant letters to, capital letters to show the dominant trait and lowercase letters to show the recessive trait. Some examples of dominant traits are brown hair over blonde hair, brown eyes over blue eyes, tall over short. These are dominant traits, and recessive traits, again, are the ones that only show if you have those two traits from both of your parents. So sometimes they can be hidden. If you are a carrier of a recessive gene, that means that you are heterozygous and that recessive gene is hiding in your phenotype because the dominant gene is expressed over it. Then we look at Punnett squares. So a Punnett square is a way of working out all the possible offspring genotypes and phenotypes that we can get from breeding two individuals together. Now, that doesn't mean that our offspring are definitely going to be all of those or one of those. These are our chances. So if we figure out all 4 possibilities for a single trait, that means that our offspring, the baby that comes out of that breeding, is going to be one of those 4. We don't know until they're born. But it is a helpful tool to show all the different combinations that can come from breeding two individuals based on the genes that we have. So figure 1 on slide 19 shows the crossing of a brown-eyed heterozygote with another brown-eyed heterozygote. They both show brown eyes in their phenotype because although they are heterozygous and they have one brown eye allele and one blue eye allele, that blue eye allele is recessive, so it's not going to show. There's some practice with working on Punnett squares. I want you guys to do that yourself. You take the genes from one parent and segregate them across the top of the Punnett square, and then you put the genes for the other parent, segregate them across the side of that Punnett square, and then you match them all together to see what all the possible combinations would be of their genes. You can see that practice in slides 20 through 28. I really want you guys to work on these yourselves, and then if you need extra help, we can do a video lesson and like a video meeting through Zoom and kind of work through these together. And your worksheet will also give you practice on those. So that is it for introductions to genetics. Basically, you need to understand what is a phenotype, what is a genotype, what is an allele, what is a trait, how are genes expressed, who is Gregor Mendel, and how did he help us understand genetics? What is dominant, what is recessive, what is homozygous, what is heterozygous, and how do you do a monohybrid cross Punnett square? That is a Punnett square looking at just one gene. After we work through all this stuff, I'll be uploading the second part of your genetics lesson on Wednesday next week. You have a week to work through the genetics part 1 assignments that I'm uploading today. So the worksheet, exit slip, guided notes, those won't be due until next Friday. Go ahead and follow the playlist that's attached to this channel and email me with any questions. See you guys next week.